So we have heard a lot about sequences in neural activity, manifolds, how they control behavior, and so on. So I'm not going to do that. I will very briefly show you some more slides that you have already seen, perhaps a little bit different. But I will go a little bit more into the networks that generate these manifolds, because this is something that is has received a little less attention. What generates these manifolds? Of course, they're very useful in controlling behavior and do all kind of nice things. So I suppose me and followed by Srijan, I think we go a little bit more into the mechanisms which might be underlying these low-dimensional manifolds. So that's the plan. So I'll spend some time on low dimensionality. In particular, I'll try to ask a question, which is that why is it so difficult to change these manifolds? And I think the last discussion that we had with Sarah, that already has the nice introduction to this topic. Then I'll move a little bit away from just manifolds and go into sequences. And finally, I'll talk a little bit about neuromodulation and how it could be a very nice way to change manifolds or the structure of neural activity on rapid behaviorally relevant timescales. I mean Sebastian and Emil. Emil is here in the audience. Emil worked on the low dimensionality part of the work that I'll present, and Sebastian worked on the sequence part. So I did almost nothing. I'm just standing for them. So this is the situation, right? So we saw this, that today we have the technology to record thousands of neurons. No issue about it. But I think we are very limited in what we can do because of our training. As physicists, we are trained to count up to 2, maybe 3, and after that it becomes infinite. So we don't really know what to do with these thousands of channels that we have recorded, and therefore we resort to something that is manageable, something that our brains are able to make sense of. And that something is either we do dimensionality reduction, linear, nonlinear, or any other form, or we search for neural assemblies, which goes back to the old ideas of Hebb and Sherrington and Lashley and so on. And I think this data is really very good for asking these questions. With single neuron or 2 neuron recording, we couldn't really ask these questions. So now we are in a very good position to exploit this multi-channel data. So what comes out of this is we have already seen what happens with the dimensionality reduction, that you transform these many units that you have recorded for different behavior. We saw some data about motor space. This is about olfaction, very old work. And I think this is one of the first ones where they try to reduce dimensionality of data and make sense of it. And here what they showed was that you can see even the stimulus-specific trajectories, and there are some fixed points, and there are these trajectories which are far more reliable than the background activity. So that's roughly the state from here. This paper was mentioned in previous talk by Sarah. I think it's an excellent reading. We all should read this. Besides everything, there is this nice summary diagram where they picked data from various studies all the way from 1999 to 2013, maybe some newer ones as well. And what they show here is that most of this data indeed suggests that the neural activity is relatively low dimensional. I put this star here. I don't know where exactly it should go. It should go somewhere behind this legend, but I put it here. This is the paper that Ken Harris will talk about, I think, on Friday. So this is saying that it's not low dimensional, but high dimensional. Sarah also mentioned that Ganguly has given us a very good way to think about why this should be low dimensional, right? We were talking about the task. The task for center-out movements is very simple. In fact, most of the tasks we ask the animals to do are very simple. So in that context, it may not be that surprising that the activity is low dimensional. Moreover, we are recording activity over a very short time. If you have n time bins, you can't really go beyond those dimensions. If you have recorded 1,000 units and you have recorded only for 100 time bins, then you can at best span 100 dimensions. So there are all these issues that we should keep in mind when we think of dimensionality. But I think work has been very carefully done that we can trust this analysis, that indeed in many tasks that are commonly used in neuroscience, it is indeed that the brain is low dimensional. So up to these things, for me, this was just a nice trick that we have to resort to because we don't know how to count beyond 3. But this paper changed this opinion. This is from Aaron Batista's lab. What they did here is they tried to exploit these manifolds and see whether they have any behavioral relevance or not. And the task they did was that first they check where the manifold is, then they design their BMI decoder, which exploits the fact that the activity lives in a manifold. So up to this time, it's good, and then they start to perturb it. One perturbation they could do is that change the BMI task such that now this trajectory no longer takes you, let's say, from left to right, but say from up to down. This they call as inside manifold perturbation, meaning you reorganize your trajectories. Other idea would be that you can actually map the task or map the activity in such a way that you assume that there is another manifold which is orthogonal to the existing one. And question is whether monkeys can learn this task or not. When I interpreted this task, to me it was like, you learn to play cricket and then you are asked to play baseball. You cannot do that. I mean, these are completely different manifolds, so it should not be possible to do it. And indeed, monkeys pretty much do that. If you do a within manifold perturbation, your performance decays when you introduce the perturbation, but it slowly recovers, and you are a bit slow, but still you can do the task with fairly good reliability. But if you go outside the manifold, it's really very bad, very slow, so monkeys find it very, very difficult to learn this task and perform this. So question to me was, why is it so difficult to change these manifolds? And it's a difficult question, so I gave it to Emil. He was a master's student looking for a thesis topic, and I said, Emil, you do this. And to my surprise, he did it, and you will see what he did. So that's all due to Emil. So when you start to ask this question, you ask, like, how do I get to a network which can have low dimensionality in the first place? So one way is that you somehow think that there are structures in it which are carrying this low dimensional manifold and so on. So we looked into the literature that how can we design a network which shows some low dimensional behavior. And largely there are three approaches to do this. One is called neural engineering framework, which exploits the fact that we have tuning curves and you can use them as a basis function and thereby create any arbitrary function. Other is efficient coding networks. Perhaps Sophie will talk in a couple of days. But there also the idea is that you know the dynamics your network has to exhibit. You use that to construct a connectivity matrix via the network, and lo and behold, you have that behavior. And finally, you can use echo state networks. You take a reservoir, you tune it, train it so that it follows certain trajectories. Doesn't matter which one you use, you will get a network structure which will have low dimensionality. Now, what was interesting that we found here is that actually all these, they have different names, but at the core of it, it's actually quite similar. If you think about neural engineering framework, or let's say force or echo state networks, so this, if we construct this system in following way, that we have a recurrent network, we read the activity using a decoding matrix, let's say phi. We create a low dimensional description of this activity in axis, then we feed it back through K. So this is now a recurrent system where we are reading the recurrent activity through a decoder and then encoding it back. This system, if we take it, we can convert it into force. We can say that this is my reservoir, and these phis are the weights that I'm tuning. And then whatever is the error that we feed back. So this is how from the same network we can create an echo state network. If you want to use neural engineering framework, then we set this omega to 0 and calculate k's and phis to follow a desired dynamics. If we want to use efficient coding network, then we don't set omega to 0, but we assume that the task omega does is that it basically makes sure that the activity remains sparse and all the behavior is encoded in phis and k's, and this gives me the desired structure that I want for my network. So this was quite interesting to find out that even though we call them as three different ways to construct networks which will perform some tasks, turns out that they share some underlying principles which are very similar. So anyway, we had a recipe then to create a network which shows a low dimensional manifold. So this is what we have as a model that we can now train to exhibit whatever dynamics we like. Now, how do we relate it to the experiments that were done by Sadler? To do that, we say that since we are reading this network through phi, these are equivalent to randomly sampling some neurons in the network that you have used to create the principal components or the factors and other low-dimensional measures. So our claim would be that in this structure, you could use these readouts and construct your low-dimensional description of it. That is the Z's, or the principal components.
So X1 and X2 would be like the modes, essentially, of the network. They are combinations.
Exactly. So right now I'm exposing them like this, but eventually they are part of one network, meaning effective connectivity is just a product of this k, omega, and phi. So you are not really familiar with them. In a way, axes are the latent modes or the latent variables of this network. So the claim is that these latent variables are nothing but the principal components that we measure. And if we can show that it is indeed true, then we can ask a question that what happens if I want to reorganize these latent variables. So first, Emil did that. He makes sure that whatever we are doing makes sense in this context of data analysis that was done by Sadler, for instance. So he trains this network to act like an integrator, then reads the activity, gets the principal components, and then we try to relate like how these principal components measured with PCA or factor analysis, how do they relate to the encoding and decoding matrices. Looks like there is a fairly good agreement. So it's not exact, but it roughly does the job. At least we have a model where we have something that we can play with. So when we do that, now let's say that we have this network, we can check whether it indeed shows low dimensionality, and it does. So we wanted it to have 4 dimensions. You can extract that. You can look at the principal components of the activity. It indeed has very big, like 4 eigenvalues that explain the variance pretty much. If you look at the spiking activity, that looks reasonable, at least in this raster diagram. We know that there are problems with this, but this is fairly good. So we have a network which has low dimensionality now. So we can do the experiment that Sadler did now. But before that, we also checked like what is going on in the structure of this network. The way it was constructed, we did not really have any control over how the synapses will come up and whether it will be sparse or not. So we had to put these restrictions that network should be sparse and there should be an EI balance. So we do have EI balance, the network is sparse, and something that was interesting that came out which we did not know or did not put in, that our matrix that reproduces this activity does not have really low dimensions. And I put a pointer here to Srijan's talk because I think he will come and he will disagree or say more about this.
Because the three learning methods that you show, there the matrices are automatically around. So how are you training? So you're training and then you're sparsifying?
No, we are not sparsifying it. So we are training it and we put this restriction that the network should be Dalian. And to begin with, we put the restriction that there should be only 20% connectivity. Effectively, we end up with less connectivity than 20%.
Is it sensitive for this 20% if you start with something else?
This I don't know if it will be sensitive, but we had so many different realizations of this, so I don't think that matters. What comes out of this training method is that the weights are log normally distributed. So that's the distribution of weights. And turns out that most of the dimensionality is actually carried by strong synapses and not by the weak. So we could then do this experiment of perturbing the connections that you delete, let's say, weak connections or the strong connection and see how this structure is deteriorated. And for weak connections, it really doesn't matter. So it may be that it looks like low rank when you threshold the matrix and only consider strong weights. This we don't know. We right now took.
I'm so confused about what training are you using? So you showed the three options. So you're doing.
So here these results are from neural engineering framework.
But there you have to be low rank.
I mean, take it?
Yeah.
You are looking a bit.
No. So, so what we do is we randomly select a number of synapses and say these are the ones that are trainable, and the other ones are not trainable. So that gives you a pattern of forcing the matrix that only some parameters are changeable. So when you look at the single value spectrum of the whole matrix, it becomes high dimensional, even though we are sort of training just a subset of the matrix, and those are randomly selected.
Good. So now we can do this experiment of Sadler. So first we do that perturbation. We go from make an inside manifold perturbation and retrain the network and see what kind of weights come out. These are the original weights and these are the new weights. So you start with some connectivity matrix, and now what you have done is change this mapping here so that it stays within the manifold. If we do that, yeah?
The mapping of the phis. You're changing the phis.
Yes, exactly.
But staying with the same.
This is for, yeah,
hyperplane.
exactly. So this is for that. And so new weights are pretty similar to the original weights. There is a small change that is happening. On the other hand, if you now do the same thing, like going out of the manifold, turns out the weights are completely off. It's as big as if we start to train the network from scratch. So this gives an idea that why these manifolds are so difficult to learn. In a way, in a retrospective way, it's kind of obvious that after all, the connectivity determines these manifolds. And if you change it, then of course, this will get out of it, right? So it's dependent on synapses then. Manish.
Is the subset of synapses that you change the same as in the first?
It's the same training method then.
And the same subset?
Yes. Yeah. Okay. So this is somehow more an experimental way of showing this. That in this model, this is what we get. So could we really understand this, what's going on? So we give you a little heuristic like why this is working this way, why you need such a big change or such a small change to stay within the synapses, within the manifolds. So if you want to stay in the manifold, what do you need to do? You have this low dimensional structure here in, let's say, in axis. So you have chosen a particular phi, which restricts you to, let's say, 4 dimensions now. And what I want is that my trajectories move within this 4-dimensional space. So we could imagine a transformation on my PCAs, let's say with this orthogonal matrix Q tilde. And this is, of course, a d-dimensional matrix because we want to stay within this d-dimensional space. So if you do that, that means that you need to transform also the phi, also k. And if you do this, because we are assuming that this is an orthogonal matrix, what follows is that the new matrix that you want is pretty much the same as the old matrix. So moving within the manifold, it seems that would not require such a big change. On the other hand, if you want to go out of the manifold, what I need to do is I need to go back into the n-dimensional space and rotate these d dimensions in that. For that, I need to do a similar kind of operation, but in this case, now we have the PCAs. We need to transform these components using this matrix Q hat, which is now n dimensions. So if you do this transformation and follow the same rule that you need to transform phi's and k's, you end up with a new matrix, which is Q hat times W times Q hat transpose, which means that the new matrix is going to be very, very different from the previous one. And that's what we experimentally also observed in these simulations. So I think this is a good heuristic to think about, that when we are staying within the manifold, we are just making a d-dimensional rotation or reorganization of trajectories. But if we want to go out of that, we actually have to move in this n-dimensional space. So I think this is a good heuristic that gives us an idea like why this manifold change is so difficult. We take a leap of faith and say that because connections matrix are so different from the original matrix, perhaps learning will be very difficult. If the matrices were similar, most likely it could be learned with relatively fast or some intrinsic protocols. So there is a jump here, of course, from the structure of the matrices and to learning. But I think it's fair to say that bigger the change in the connectivity matrix, longer it will take you to learn. So this we can show also for a spiking version of this model, not just for the continuous rate models. And again, the same thing that you have, if you make inside manifold perturbations, the new matrix is very similar, outside matrix is not so. And in fact, you can start with pretty much scratch and it will be still the same. Then we can use this and go beyond what Batista and Byron Yu did. And we can ask a question like, what other perturbations exist, perhaps even within the manifold, that may be easy or difficult? So what we do there is that come up with, this is their perturbation, that to stay within the manifold, they permute the principal components. But you can also rotate them by 45 degrees. You can choose a matrix for transforming them, which has numbers drawn from a Gaussian distribution. Or the matrix could be like identical elements and so on. So in all cases, your learning is better than outside manifold. That will be somewhere here. X-axis is, so this just tells you the correlation between the original matrix and the new matrix that you want. But indeed, there are some perturbations that are going to be very difficult, for instance, those with same elements. Some of these, I think, were tested in a recent paper by Byron's group. So this appeared pretty much the same time when our paper appeared. So it looks like that this heuristic kind of works even for those. So based on this, I think we can say that manifolds are a consequence of the connectivity matrix that you have. And in this case, we are explicitly assuming that this connectivity matrix encodes the task the monkeys have been trained to do. Obviously, these are heavily trained monkeys, right? They have done, I mean, this task they have done like thousands of times, as we saw. So maybe this is overtraining, maybe not. But at least to the extent the task is encoded in your matrix, changing manifolds would involve a massive restructuring of the connectivity, and therefore it is likely to be very difficult. What we also kind of learned here that if you knew the full connectivity matrix, which will be a product of k, omega, and phi, that wouldn't tell you much. Meaning knowing the connectome perhaps is not so useful. What Sarah was saying is far more interesting, because looking into these latent variables or the low-dimensional manifolds, I think, can give you a better information about the task structure as opposed to looking at the full connectivity matrix. But this is just a side remark. Anyway, so this is one aspect of multi-unit or multiple single-unit data that we are working with. Other aspect is sequences. So besides doing dimensionality reduction, there is another way to analyze this, which is to look for sequences. And we look for sequences, I think the tradition goes way back. There is this nice line that appears in a book by Sherrington, where he talks about how a brain is supposed to represent behavior. And he basically associated the brain to a loom, perhaps because he was, maybe he grew up in this industrialized UK, where the most sophisticated machine perhaps was the loom. So he associated the brain to a loom. Today we do it to a deep network or something like that. But I think this is much better. I like it better than the deep net connection, because there is a dynamics here. Dynamics in the sense that he talks about an enchanted loom where millions of flashing shuttles weave a dissolving pattern, always meaningful, never an abiding one, a shifting harmony of patterns. Meaning whatever task I need to do, if I need to move my hand in a particular trajectory, if I need to throw a ball, it should require some sequential activity somewhere in the brain. At least one brain area should have sequences. And those sequences should not be abiding, meaning persistent, but should be task-dependent, context-dependent, and so on. And the same sentiment is expressed in Hebb when he envisions his phase sequences, that this is how behavior is represented in the form of sequences of activation of neurons. So since then, there is this idea of neural assemblies and how neural assemblies concatenate to create meaningful behavior. And now, because we have so many units, we can record it. This was already known for hippocampus, where you see the sequences of place cells, not only when the animal is running, but also when animal is planning to run or maybe remembering his previous runs. Meaning that these sequences are not just driven by the external inputs, as perhaps you would imagine in these cases, but they are also internally generated. Still, in this task, there is a very strong motor component, but you can also think of tasks where there is a very strong cognitive or memory component, like this experiment by Chris Harvey's lab, where they look into an activity in parietal cortex, I think, and see like when the animal is making correct left choices or correct right choices. The idea these days is that earlier, people would take these neurons, sum their activity, and show us a PSTH. And what you will see is a persistent activation. Now, because we have hundreds of neurons, in this case recorded using calcium imaging, they try to organize them according to the time when they reach their peak activity. Now, this is maybe arbitrary, I don't know, but it gives us a nice result that if you organize it, you see a sequential activation of different neurons, and the sequence somehow is selective whether you are going to the left or to the right. So in this case, one could be fairly certain that this is not created perhaps by the external stimuli, but maybe internally, and the representation in the brain of this task are in the form of some sequential activation. So question then is that where are these sequences coming from? Could they be learned? Could they be innate? Or is there something else? Obviously, whenever you have a sequence, you would imagine that there is an effective feed-forward structure. Not necessarily a SynFire chain, but something similar. And there are many ways to do this. In fact, you can hand-wire a structure and say that, well, somehow magically it appeared in my recurrent network. Or you could train it, like what Kanaka Rajan did. So she used the force approach, fixed some synapses and trains others, and then you can get a particular sequence in this network. Other idea comes from dynamical systems, where you can use an attractor which is unstable, so that you activate it and the activity then leaks into another attractor. All these are somehow supervised. There are some ideas how you can do it in unsupervised way, but usually it will end up like switching on and switching off the plasticity to the very least. So what we were interested in is that are there some more biologically plausible ways where we don't necessarily have to train the network, but start with some generative rule and we end up with a network that gives us sequences. So what we started with was spatially connected networks, which are very well known to give rise to stable bump attractors, meaning some neurons become active and this attractor either remains stationary or it shows traveling waves. And the key to that is the so-called Mexican hat connectivity, which is that a single neuron connects in its neighborhood, and the effective connectivity, when we sum up excitation and inhibition, it looks like a Mexican hat. And typically these networks are homogeneous and so on. So we said, okay, let's try to destabilize these bumps and see what happens. So the change we introduced was that the neuron is not connecting in a symmetrical Mexican hat fashion, but that it has some preferential connection in a given direction. Now, some meaning about 1% or 2%. So out of all these few thousand connections this network is sending out, only about 1% or so are going in a particular direction. Then the question came up that because this network has space, how are you going to distribute this preferential direction of each neuron? Is it going to be random for everybody? Is it going to be same for everyone? Or there is something in between? This will be the case of homogeneous, where every neuron projects in same direction. This will be the case where everybody has a different preferential direction. And in between these two is this spatially correlated noise. This is called Perlin noise because Perlin formulated it for some Disney movie and won an Oscar for it. So I think it's a fairly good model. But anyway, it's a family of gradient noises that you can create. The good thing is that it gives us a nice way to control these spatial domains. Now the idea is that all the neurons lying, let's say in this yellow region, they are projecting in one direction. All the neurons in blue region, they are projecting in another direction. And obviously this is a continuous scale, so we have a continuous change in the direction of neurons. What comes out of this is the following. So when we have stable bumps, meaning when we have Mexican hat, then you have these stable bumps, so grid patterns or whatever you call them. If you randomize the directions, nothing much happens to these grids. They remain stable, a little bit more bumpy or more jittery. But if you connect them all in the same direction, then you have a beautiful traveling wave. So all the bumps seem to move in one single direction. As opposed to this Perlin case, where if you now watch it, that in every given observation window you will see a sequence, but quite independent of other in terms of its direction. The speed is all the same in the network, but in any given observation area you will see these bumps moving in different directions. Now this is of course the spontaneous activity of this network, but it only shows you the repertoire this network is capable of. If you stimulate it, I'll show you in a minute, if you stimulated one particular area, you will only see one sequence. This is what comes out. So if whether you take a purely inhibitory network or an excitatory inhibitory network, doesn't matter. The rule is the same, that take your neurons, give them some preferential connectivity in a given direction, and make sure that the neighbors have roughly the same direction. So that's correlated anisotropy, if you wish. So the whole network is anisotropic, but neighbors are similar. And if you do now the experiment of picking up some neurons, like behaving as if you have put a Utah array or you are doing calcium imaging, you are locally recording or scanning the whole network, depending on that, you can get these kind of sequential activation patterns. So it's actually quite a simple way to get connectivity in a network which is sufficient to give rise to sequences. Now, obviously, if you want to measure dimensionality of this, if you picked up any particular region here, neurons are getting activated in a sequential fashion. So if you did this standard PCA, you will end up with a very high dimension. So Jonathan was right in the morning when he said that for sequences, it may be better to use something that he introduced, the nonlinear way of extracting the dimensionality. And if you did that, here it will be really, really low dimension, maybe 1 dimension, for instance. But also it depends whether you scan the network locally or globally, picking random neurons or some close neighbors. Depending on that, your dimensionality will change. Space, number, or stimulus that you put in. Also, it's not that there is some threshold for these sequences to emerge in this network. We have one extreme where all the angles are randomly distributed, and here everything is homogeneous for everybody. Here we know that there are no sequences, and here, if you wish, we have one sequence. And there is a nice continuum. As you change the scale of these correlations, you go from more sequences to fewer sequences, but those fewer sequences, they can travel longer distances or they can last for longer. So that's quite nice that on one hand, we have many sequences, but they don't really move. On the other hand, we have very few sequences which move very rapidly and persist for long distances. So in a way, we can just tune these connections and get different kind of sequential behavior. And this is what will happen if you, as I said, this was spontaneous activity. You can create nice persistent sequences. If you just evoke activity in some neighborhood, it will create a reliable sequence every time you activate it. Of course, if the background is strong, then you will not be able to recognize what is happening. The orange dots are the evoked sequence, and the black dots are the ongoing sequence, so there is no real distinction. That means that network has to be relatively silent to see this. And we can, of course, parameterize this, that depending on the input parameters, where exactly is this sweet spot where you get the sequential activation, where the response time is maximum or minimum, and how reliable these sequences are. So in a way, by controlling the input, once you have put this generative rule for wiring the neurons, you will end up with a network which exhibits sequences, and the way you measure its dimensionality, you will end up with a low or high dimensional manifold. And the reason it works, I think the way we are wiring it is that, first of all, if you look at the eigenvalue spectrum of this network, turns out as we make these preferential connections more and more, meaning we are changing the space, so more and more neurons are projecting in a single direction. Eigenvalues, they leave this central lobe, and for a homogeneous network where we have beautiful traveling waves, there these eigenvalues are really big. Otherwise, in between, it just goes from here to here in a nice continuous way. So these eigenvalues that are leaving the central lobe, these are the large ones that you will pick up as your PCAs. So in a way, the number of eigenvalues that are leaving the main lobe, that is a determining, or that's kind of the dimensionality of this model or this network. The next thing we did is that we did a kind of anatomy on these networks. And what we did is that we went into some location, try to find out if we can find a feed-forward path corresponding to a sequence. And of course, we do find. If you have everybody projecting in the same direction, you get many, many paths, very long. But if you are in the Sperling mode, the green traces, you get fairly large, very wide distribution of effective path lengths. So by making this correlated anisotropy, what we have done is unknowingly, inadvertently, we have embedded a lot of feed-forward paths in this network. There is no particular arrangement, but they are bound to appear the moment you wire it like this. Now, this is nice in a way that it gives you a very simple rule to create networks which will show structured activity. Question is that, is this good enough? This, I don't think is good enough. It just tells you a way to do this. The problem with this whole idea is that this is all fixed. I choose the connectivity, I decide which angles neurons will go, and if we are in a biological situation, then once I have decided the axonal or dendritic arbors orientation, I'm done. That means that these networks are actually, even though rich, but completely boring. They have many sequences, but these are the same sequences they will exhibit. So this was not nice. So we thought that we really don't answer this question of sequences, because what we have is essentially abiding sequences. They all appear again and again in the same fashion. How do we get out of this? How do we make it a dissolving pattern which repeats in terms of task and contexts? For some other project, we were working in understanding the spatial arrangement of dopamine. This was related to Parkinson's disease, nothing to do with sequences or anything. So what we were looking in is that if you have a dopaminergic neuron, its axons are going to release this neuromodulator, how the neuromodulator will diffuse in the space. Naively, of course, we will start that this is going to be a diffusion, isotropic medium, but this is not true because brain is full of all kind of obstructions, blood vessels, glia, neuron structures, and so on. So if you now assume that these black dots are the axon terminals which are going to release the neuromodulator, and assume that there is some anisotropy in this medium, immediately, even though the release is pretty predictable and deterministic and uniform, you will end up with local patches with high and low dopamine. Meaning that by default, because of the structure of this tissue, you will end up with a spatially inhomogeneous distribution of the neuromodulator. Then this paper came out where they have now started to image dopamine, in this case dopaminergic receptors in motor cortex, and it turns out that even here, if you look at it, the distribution is rather patchy. So this notion that there is a phasic level of dopamine or tonic level of dopamine that often we think of, like something floods or maintains a baseline level, that's not true. There is a fairly high degree of inhomogeneity in these distributions. What can we do with it? We know that neuromodulators, they change neuron excitability or they modulate the synaptic strengths. Okay? So let's assume that I have a particular neuromodulator which is going to change the strength of synapses in a consistent direction, whether it increases or decreases. And let's now not wire the network the way I wired it, meaning there is no preferential connectivity whatsoever. Wire it in a fairly homogeneous way, and then go back and release neuromodulator substance. And obviously, this will be a bit patchy, given the anisotropies and the axon distributions. You end up with these clouds of high and low concentration of dopamine, or whatever your favorite neuromodulator is. Wherever this is high, these connections will be strengthened and others will either be unchanged or maybe renormalized and perhaps weaker. That means that essentially along the gradients of this particular substance, you will end up creating effective feed-forward pathways. And these are the pathways that essentially we need either to create sequences or to create a predictable and low-dimensional manifold. So the idea then is that such manifolds could exist, and they could be created transiently because of release of some neuromodulatory substances. And as the pattern of this substance changes, you may end up with completely different sequences. Same neuron can participate into many sequences, and they can change on behaviorally relevant timescales. So this is just to give you a cartoon of that let's say in situation one, this is the set of neurons that release the neuromodulator, they are activated, they create a patchy arrangement, and as a consequence, you have this set of temporal sequences that may arise. Task changes, or the attention changes, or the context changes, a new set becomes active that gives you a somewhat similar but still different pattern, and that will create functionally, or as long as this modulator substance stays, a new pattern of sequences, and so on and so forth. You get a fairly good reliability if it turns out that the reward system is the same, like in this monkey's case, that you move the task, you get the same reward, you have all the same setting. So most likely similar neurons are being activated, similar pattern of neuromodulator is created, and therefore you have, no matter what neurons you read, in every trial, short period, long period, you end up with similar kind of dynamics. So I think this is a fairly cheap way to create reliable functional circuits without any supervised or unsupervised learning. What you need is some kind of a generative rule for this. So just to summarize this now, that of course the core of these manifolds and so on is the connectivity. That connectivity could reflect your task, the context, or the animal's learning history. If you want to change it, if you want to do it completely drastic, like you have spent your time learning cosine-based receptive fields, and suddenly you are supposed to work with manifolds, of course you need to redo everything. You need to relearn a lot of things. And that will require a complete change in your connectivity of the neurons, and therefore you will have a resistance for that. But I think what can do a trick on a large scale, how you can rewire the networks, actually not rewire, but transiently in a context-dependent way, how you can modulate the connectivity without any plasticity rule or so, is through neuromodulators. And neuromodulators looks like this is quite natural for them to create an anisotropic patchy arrangement of release. And therefore, along those gradients, you will end up with functional circuits. And as long as the neuromodulator release patterns are reliable, your behavior will be reliable, doesn't matter which neuron you pick up, and so on. Cool. So that's what I leave you with. If you have more questions.
I would like to get an intuition. At the very beginning, you gave us this very nice argument of why these three ways of thinking about this type of networks are actually different ways of thinking about the same network. But then you said when you analyze this network, you get the matrix omega, I understand, is not low rank.
No, matrix W is not low rank.
Well, the matrix W is a connectivity within the omega, isn't it?
No.
Then I misunderstood.
So, I'll just repeat then. Okay, I can stay here. So this is our full network.
Right. And W is the connectivity of the full network.
Yeah.
Including K, omega, and phi.
Exactly.
Okay. So you said it's not low rank. The weights have this log normal distribution, and each neuron ends up being excitatory, inhibitory balanced.
Yeah.
And I was trying to get an intuition of how you get excitation inhibition balance out of the requirements that you impose on the network, if there is any simple way of thinking about it. I understand that it comes out, but...
No, I think the reason it comes out is that we have a restriction on the firing rates of these neurons. These manifolds are, or the PCAs or factors are calculated on the firing rate, like using these spike trains. So I think the reason it turns out to be EI balance is that there is an upper limit how much firing rate we can have for neurons.
And the only way to achieve that is with EI?
I don't know. This is how it turned out. Okay, so we unfortunately don't really know so much about these structures, right? Because we train them, we don't really get this synthetic feeling about it.
It's difficult to do the autopsy and understand what was the underlying mechanism.
Exactly.
Okay.
So, hi, Arvind. So, you mentioned dopamine. Of course, my ears pricked up immediately. So, your idea there was really nice, that you have the idea that you want some neuromodulator release that's really heterogeneous, this patchy release, to drive the local synaptic connectivity profile, basically, to couple the neurons into chains.
Yeah.
But your point there was different sets of patchy release would cause different chains.
Yeah.
But how would that happen? So the patchiness is driven by the random clustering of the release sites, and that's always the same.
Yeah.
So where would the different profiles come from?
I'm going to give a circular answer to this because I don't know the full answer. So it turns out that dopaminergic neurons themselves are tuned. Right? Exactly. So I also do the same. So the big requirement would be that some sparse or weak tuning of dopaminergic neurons or some variability in those patterns. Otherwise, there is no way. Otherwise, you are stuck in the same way if the same set is activated all the time. So you need some variability there. I'm saying circular because I remember asking this question to Elena Witten because she's the one who shows that there is some tuning in dopaminergic neurons.
They are tuned to what when you say they are tuned?
They are tuned to some task phases.
Temporally.
And then I was asking, so I mean, it's such a small network and they are not directly getting sensory input. How are they becoming tuned? So this is, of course, not really very well clear and understood. So unless there is a tuning, this idea will fall apart, obviously.
So doesn't that make a prediction then that there should be some spatial scale on which you're seeing this modulation?
Yes. So also, not only the spatial scale, so these models will also make a prediction how long this modulator substance should stay, what should be the dwell time. Because if the modulator stays too long, you will create a runaway, or these functional circuits will become somehow looping on each other and it will explode. So you have to remove the substance relatively fast as well. The way the whole thing, I think, wraps around is that, so Rui Costa always talks about dopamine release when you initiate actions, self-initiated actions. So there looks to me that as if the first pattern of the release would initiate these circuits, and then you will perform the task. Maybe you get a reward or not, so you'll get a second boost. And depending on that, you will reinforce some of them and others will die out. So there should be some selection mechanism on these. So it works both motivation as well as learning signal.
More questions?
Okay.
Oh, sorry.
Well, can one extend this? See, what I'm fascinated about is to have some network that actually maintain its activity without running into excitation oscillations and so henceforth. So could you modify, say the dendritic tree of neurons and make an inhomogeneous, shall we say, maybe even with random connectivity that will sustain its activity? Because I think we need these kind of network models actually to really play with this, because the most of situations are an interaction between some intrinsic activity in the brain and some minor outside perturbations.
Mm-hmm. So these networks, they do have this mode. We have to tune certain parameters that they become spontaneously active and generate, let's say, quasi-periodic. I wouldn't say they are chaotic, but they are quasi-periodic activity patterns. Can't be sure about this because, of course, we have a finite network and we have to wrap around and so on. So can't be really sure about the full temporal dynamics of it. But over a short period of time, you can easily create self-sustained activity. Like this is a nice example, right? In this case, of course, it dies out. And we definitely don't want a situation where stimulus evokes an activity and that sequence just runs around for infinity. But obviously, if you change the EI balance in this network, then you activate the sequence and it will live forever. Or, I mean, not forever, of course. Yeah.
And react fast to small external perturbations, for example.
Yes, so then we need to understand better like how these ongoing sequences interact with stimulus evoked sequences for sure, yes.
Thank you very much.
Great, thanks.